Bismillahirrahmanirrahim. Assalamu alaikum. I am here from Conceptual Medicos, and today in this lecture we will be talking about the Bordetella pertussis. As we know that there are three genera related to the respiratory tract. The first one was Haemophilus, which we have discussed in the previous lecture. The second one was Bordetella, and the second one is Bordetella, and the third one is Legionella. So today our main topic is Bordetella, and especially the species Bordetella pertussis. Okay, so coming to the features of pertussis, we know that they are gram-negative rod related to respiratory tract. They are coxo-bacillary by shape, means their shape is somewhat between cocci and bacilli. They are encapsulated, but this point is not very much important because their capsule is not a virulent factor. They are strictly aerobe, which means they can do the which means they are oxidase positive. Now the transmission. So these are transmitted by the airborne droplets. When a patient of the whooping cough caused by the Bordetella coughs, then he transfers the organism through the air droplet. This is a very highly contagious disease and means the organism can easily be spread from one person to another person in the community. Coming to the epidemiology, so it mainly occurs in infants and in young children. In adults, its incidence is very low. The cases have gone down due to the vaccination. The outbreaks have occurred due to Bordetella pertussis in 2005, 10, and 12. Coming to all the pathogenesis, which is very important of this organism, so several virulence factors are there for pathogenesis. The first one is filamentous hemagglutinin. Let's suppose this is bacteria. These are villi on the surface of the bacteria. These villi have a protein known as filamentous hemagglutinin. Let's suppose this is respiratory tract, the respiratory epithelium, and these are the cilia. These proteins known as filamentous hemagglutinin on the villi are able to attach the bacteria to the cilia. So the cilia becomes less motile and the symptoms of the disease appear. So the filamentous hemagglutinin helps in attachment of the bacteria to the epithelial surface of the respiratory tract. The second one is the pertussis toxin, which is the important one among the virulence factors. This pertussis toxin has two subunits, A subunit and the B subunit. The B subunit is the binding subunit and the A subunit is the active subunit which will perform the action. When the B subunit binds, the A subunit is inserted inward. Note that this A subunit will do ADP ribosylation of the G inhibitory protein, which will inhibit the function of the G inhibitory protein. So when G inhibitory protein is inhibited, it will cause increased level of adenylyl cyclase, which in turn will activate CAMP, means it will cause the ATP to convert into the CAMP. This CAMP will cause protein kinase to be activated. These protein kinases will then phosphorylate the ion channels, and when these ion channels are phosphorylated, there will be a rush of ions from inside the cell to outside the cell, followed by water, and hence edema of the respiratory surfaces will occur.